It's great to see everybody. Gosh, wow.
See, advantage, right? Advantage to
doing
it this way.
Yeah, crazy. So.
Okay, great. We are streaming live on YouTube, and we'll probably just give people another minute or so to keep joining in, and then Adam will take it from there.
I'm dealing with the late flurry. Every week there's like 10 people who email saying, can I get the link?
I'm always surprised that we keep getting signups just like consistently, like a few more people every day coming on the mailing list. It's great to see so many people interested in cell migration.
Indeed.
Okay, I think all my symbols are done. So maybe we can get started as people trickle in. So hi everyone, happy Easter. So today we've got our speaker, Jim Baer, who is a professor of cell biology and physiology at the University of North Carolina at Chapel Hill. As a graduate student, Jim discovered the founding member of the ScarWave family of ARP23 complex activators. And since starting his own lab in 2003, he's continued to study the regulation function of ARP23 generated networks, as well as other actin binding proteins. His lab's also interested in directional cell migration, particularly chemotaxis and haptotaxis, as well as trying to understand invasive tumor behavior. And today he's going to talk to us all about ARP23 and lamellipodia. So feel free to take it away, Jim.
Thank you, Adam. And thank you so very much for this wonderful invitation. You would think after 14 months, I would be better at Zoom, but okay. Can everybody see my screen? Thumbs up? Yep. Okay, great. Yes. Okay, so it is a real pleasure to be speaking to cell motility, cell migration aficionados from all over the world. And I figured if you got to suffer through another Zoom call, at least attempt to be entertaining. So, what are lamellipodia for anyway is sort of my title, and it's a structure that I've been fascinated by for a very long time. And actually in a happier version of the 2020 timeline, we would all have been celebrating the coining of the term lamellipodia, and by this guy, Michael Abercrombie. The way he described it was wonderfully British: a thin, sheet-like, mobile, commonly transitory projection from the cell. And we are still studying that 50 years later. And what I'm hoping to do is sort of tell you a little bit about what we've discovered about the function of lamellipodia and how it works. And so what you're seeing on the right is a movie, a 3-color live imaging movie of various factors that we've tagged, and I'll get into more of the details of that in a minute. But if you were to take the plasma membrane off of one of these structures, these lamellipodia, you would end up, so Michael Abercrombie also, I have to say, really gave us a very nice understanding of cell migration with his so-called 4-step cycle. And so the 4 steps of cell migration, as we understand them, are sort of, again, protruding one of these lamellipodia, getting that lamellipodia tacked down, squeezing the cell body and all of its contents forward, and finally letting go of the adhesions that you made in step 2. And this has been a very, very useful framework for understanding the various sub-processes of motility. But as I'll argue at the end, I think that in some ways this framework is holding us back from a broader understanding of cell migration. And we need to sort of take the training wheels off, as it were, so that we can begin to understand some of the more complex issues about how these so-called steps relate to each other. But again, I'm pretty invested in the steps, and the step I've been most focused on is lamellipodia. Again, if you were to strip off the plasma membrane, as Tanya Svekina and colleagues have done for many years, and look at what's underneath, the engine is actin, and it's a very specific kind of actin. It's this dendritic branched actin network. It's dense, it's dynamic, it's moving backwards from the edge towards the cell body in a process called retrograde flow. And it's been the subject of many, many papers over the years. And in fact, Tom Pollard and Dyke Mullins came up with a very famous conceptual model called the dendritic network model, where they were able to map many of the components that we know localized to the leading edge into some kind of sensible series of events that happen, starting with the generation of the branches. And the key factors, of course, the ARP23 complex that I'm sure many of you are familiar with. It's a 7-protein complex. It's nearly 1 billion years old. I'm going to talk a lot about ARPC2 because it's there's sort of our handle on this particular complex, but that's just one of the 7 subunits. ARP2 and ARP3 really constitute the first 2 subunits of what's called the daughter filament that branches off of the side of the mother filament in this characteristic or stereotypical approximately 70-degree branch. And that's really the sort of underlying basis of this. So, ARP23 is on its own not particularly active for making these branches. It requires these nucleation promoting factors, some of which I've worked on. I'm sure many of you work on them. They are themselves subject to regulation by things like small GTPases that I'm sure many of you work on. I've been a bit more focused in my lab on the sort of other side of it, which is the sort of negative regulation of ARP23 and the tuning of its activity and how branches that are once formed get preserved or taken apart by various debranching factors. But at the end of the day, the ARP23 contributes to many of these cellular processes like cell motility, phagocytosis, and vesicular trafficking. But I want you to, for a moment, to sort of do a little thought experiment. What would happen if that were gone, right? What happens to the upstream stuff and what happens to the downstream stuff? Now, it's not hypothetical because we did it quite by accident, I can assure you, a few years back. We were doing essentially knockdown and replacement of one of the subunits just so we could make a tag cell line. And we realized that mammalian fibroblasts are at least partially viable without the ARP23 complex, which was a bit shocking because yeast cells and dictyostelium cells are certainly not viable without it. But apparently mammalian cells are complicated enough that they can kind of eke out some kind of existence without the ARP23 complex. It's not a good life, I can assure you, but it's, we can at least do experiments with them. These cells lose their lamellipodia, as one might expect from the central nature of the ARP23 complex. And what it's replaced by are these really abundant filopodia. And those turn out to be really the key to understanding the residual motility of these cells. What seems to happen is the cells make filopodia without ARP23, and then they fill in the gaps between them. And they don't just fill in a convex edge, they actually sometimes have filopodia that emerge at their tips and make holes that fill in. And we really still don't fully understand all the topological problems that this introduces, but it is basically what's left. That's the residual motility. It's not particularly efficient relative to ARP23 based motility, but it does work okay to get the cells around slowly. And so, but it introduces lots of interesting questions. If you can make a cell without ARP23, maybe we can really begin to understand what they need that specific kind of branched actin for. So we've moved on from transient knockdowns to complete knockouts. We have a conditional knockout mouse in this ARPC2 gene. This is work pioneered by Jeremy Roddy when he was in my lab, and we can generate all kinds of cells from these mice and conditionally delete the ARPC2 subunit, which turns out to be, unlike a few other subunits where there are multiple genes that encode them, there's only one ARPC2 and it wipes out all forms of the ARP23 complex. The rest of the complex falls apart and degrades when there's no ARP23 around. So this truly is a complete knockout. There's no partial complex that's formed. So when we make our LifeAct movies of the fibroblasts from these cells, you can see, indeed, the lamellipodia are completely gone. The cells are spiky and they're replaced by filopodia. And I'll show you lots of movies of these things going forward. But one thing to notice is that the overall actin system is quite a bit less dynamic without ARP23. So that's an interesting question that we haven't gotten to yet. But how does the actin turnover mechanism sort of tune itself for less actin being generated without ARP23? Again, many, many questions come from this work that we're still trying to tackle. But one of the questions that I've been interested in, as Adam alluded to, is directional migration. We had been using random cell migration as really a readout for things that actin cytoskeleton perturbations, but we really have for a long time wanted to look at how cells respond to cues in their environment. And we've slowly worked our way through some of the assays to do that, especially in fibroblasts where the gradients have to last a long time. But now, again, we're in a position to ask this question. If there's no ARP23 and you expose cells to a gradient of something, can they actually respond to it? So there's three main forms of directed migration that fibroblasts at least are exposed to: chemotaxis, which is a soluble chemical cue; haptotaxis, which is a surface-bound chemical cue. I'm mostly going to be talking about extracellular matrix, but in theory, other things like chemokines could be immobilized on the cell surface, and that would still qualify as a haptotactic migration. And then durotaxis, which is substrate compliance gradients, and we're also working on that, but I won't have time to talk about that today. And so it's really quite a remarkable problem, how cells sense and respond to these various cues with directed migration responses. I'm mainly going to be focused today on haptotaxis because that's where lamellipodia really form a major, are really quite important. So the way we generate gradients in the lab is using microfluidic devices, and this is one that we borrowed from Sarah Heilshorn's lab. It's published back in 2008, the so-called Shamlu chamber. We can generate gradients of either soluble factors like PDGF or surface-bound factors, in this case fibronectin, as you'll see throughout the talk. And regardless of the cue, what we do then is we make live cell movies, just like Michael Abercrombie did in his experiments. And in this case, we're making movies that last somewhere between 12 and 16 hours because these are slow moving cells. We take those tracks then and we can transplant them so they all begin at the same place, and we can watch what happens. In this case, most of the cells end up going up the gradient, which by convention is up on the screen. And we can analyze these tracks for things like velocity and persistence and a property called forward migration index, which is kind of a blend of the directionality and the persistence. One of the shorthand ways that we often present this is using a wind-rose plot. And when you see an asymmetric wind-rose plot like that going up, that means the cells are directionally migrating. We have also more rigorous statistical tests of that. But this work is published, so I'm going to go through it somewhat quickly. ARP23 is absolutely required for haptotaxis. These are two, either a wild type or an ARPC2 knockout fibroblast plated together in the same chamber, same fibronectin gradient. Wild type cells are making their way up the fibronectin gradient. The ARP23 knockout cells are moving slowly, but completely randomly. Now you would say, well, that's kind of boring. ARP23 cells are really messed up. But in fact, when we put them in a gradient of platelet-derived growth factor, an RTK ligand, in fact, the ARP23 knockout cells can chemotax, which is, has been a challenging result for us because it goes against a lot of the dogma in the field. But we've challenged ourselves and our experiment as many ways as we can think. And other labs have begun to verify this, that ARP23 really isn't totally required for chemotaxis. Now, this is actually all I'm going to say about chemotaxis today because it's a story for another day and it involves myosin regulation, and we're still working on it. But I want to sort of circle back to this haptotactic result because that's really where ARP23 seems to play a key role. So one of the things we wanted to know, ARP23 is involved in lots of stuff. Is it truly lamellipodia? Can we probe this more specifically? And so right around the time we had this result, a new technology was coming on board in our lab called cellular optogenetics. And this is a collaboration with Brian Coleman's lab where we can activate endogenous RAC GTPases, and we can actually trigger lamellipodia wherever we want. And so what you're seeing there is the recruitment of the DHPH domain of a RAC-specific GEF called TIM1. And where we recruit that RAC-GEF fragment to the membrane, we activate RAC and we get a lamellipodia. Now, what happens if you do that experiment on a fibronectin gradient? And it was quite dramatic. In fact, the protrusions going up the gradient went quite a bit further than the protrusions that we triggered going down the gradient. So again, there's something about, activating RAC clearly triggers protrusions in both places, but those that are aligned with the fibronectin gradient are way more efficient and go further than the ones projecting down the gradient. So, this and a lot of knockdown and pharmacological experiments has sort of led us to this working model of haptotaxis, where we know sort of the skeletal outline of this pathway that involves integrin activating both RAC and SRC-FACT signaling, which we think converge on the scar wave complex activating ARP23. But we think when a random lamellipodial protrusions are happening all the time, and when they happen to align with the gradient, in other words, they go up gradient, we think that there's a positive feedback loop in signaling and protrusion machinery that gets engaged, which essentially makes that cell move a little bit more efficiently up in the up gradient direction. Interestingly, again, ARP2/3 knockout cells that have abundant Fibroblast are not sensitive to these fibronectin gradients. And what that implies is that at least Fibroblast by themselves are not capable of transmitting or perceiving those signals. I will say that one caveat here is that this is clearly a functional pathway for ECM haptotaxis. We've tested it for laminin and vitronectin and other ECM ligands, but it's not clear that other immobilized signals, such as substrate-bound chemokines, actually use exactly the same pathway. They are probably using their own surface receptors, and I think going forward, it'd be quite interesting to see how similar or different those kind of haptotactic decisions are. But at least for ECM haptotaxis, this seems to be sort of the basic pathway that we're talking about. So we've also tackled ARP2/3 function in macrophages. And one of the nice things about having a mouse, and we can make bone marrow-derived macrophages from long bones of that animal. These are innate immune cells. They're highly responsive to extracellular cues, but their real main job is phagocytosis, which uses a structure called a phagocytic cup, which is fairly related to lamellipodia. So we did some scanning electron micrographs of these cells, and they're quite beautiful. The ARP2/3 knockout cells, their sort of flat lamellipodia and flat ruffles are replaced by a series of filopodia and blebs, which is actually somewhat different than the fibroblasts. Fibroblasts, we never really saw blebs. But in macrophages, for sure, loss of ARP2/3 does something to the cortical integrity that leads to blebbing. So again, some interesting cell type differences here. So what about phagocytosis? We've sort of mainly focused on these two types of phagocytosis. There are many phagocytic receptors, but the two main types that most people study are the Fc gamma receptor, which perceive antibody opsonized pathogens and particles, or CR3, or complement receptor mediated phagocytosis, which actually uses an integrin, which you'll see in a moment, we think is important. So the assay that we've done is quite standard. We have a double labeling experiment with and without permeabilization. We can tell whether a particle is bound to the surface or fully internalized by differential labeling. And what we've done is challenge our cells, our macrophages, with 3 different size beads that are opsonized in different ways, either 2, 6, or 10 micron polystyrene beads. And one of the things that was quite surprising was that Fc gamma-based phagocytosis, which we actually fully expected to be ARP2/3 absolutely requiring ARP2/3, was sort of defective. But if you give the cells long enough, as you can see at these 60-minute time points, certainly with small beads, they're almost completely normal in their phagocytosis. It's slow. It's a little bit like the cell migration story. But given enough time, they can essentially still fully engulf these beads. Now, at 10 microns, they're defective, even at 60 minutes, but those are quite large beads. You can see one knockout cell here engaging 2 different 10 micron beads. That's a pretty tall order for any macrophage to tackle, and it's pretty amazing that they can do it at all. But it's not fully broken. So we took a look at the scanning electron micrographs of these cups, and we got really excited because it looked like the cells were using kind of a blebbing-based cup mechanism. And we thought, oh man, this is great. We've discovered a whole new form of phagocytosis. We had sort of dreams of science papers dancing in our heads, but there's a really great way of getting rid of bleb. It's called blebistatin. Blebistatin, which is the myosin 2 inhibitor, completely wipes out blebs and leaves only the filopodia. But sadly, it does absolutely nothing to phagocytosis, at least in our assay in our cells, either in the knockout cells or in the wild type cells. So I don't think it's the blebs. It's still actin dependent. We can kill it with actin blocking drugs, but we don't yet have a full explanation for what's going on in the knockout cells. Now, when we switched to the other kind of phagocytosis, we got a much more dramatic phenotype. This is CR3-based phagocytosis, and you can see a wild type versus the ARP2/3 knockout. The ARP2/3 knockout, the beads bind to the surface, and they maybe get tickled by a filopodia or 2, but they really do not get engulfed efficiently at all. And the quantification really bears that out. And that is that integrin-dependent phagocytosis is much more significantly impacted by the loss of ARP2/3 than Fc gamma-based phagocytosis. So what's next for us? To go further in this project, we needed to build new reagents. And this is where a postdoc named Mitch Butler in my lab came in and started utilizing CRISPR-Cas9 technology to tag the endogenous ARP2/3 complex, in this case in fibroblasts. So we've generated a biallelic knock-in of M scarlet into the ARPC2 locus. There's no untagged ARP2/3 left in the cell line. It's clearly pretty normal looking, and It's basically using all tagged ARP23. And this allows some really fascinating experiments, as you'll see. It allows us to really track in live cells ARP23, which up until now has been very, very difficult. So the other technology that we've sort of adapted for this project is surface patterning of fibronectin. This is a method that was worked out by Matthew Peel and colleagues several years ago. It works very well. We can make any kind of pattern of fibronectin that we want, and we started putting our cells onto them. So the first thing we did was we realized that our cells would spill over from the pattern onto the non-adhesive area around it. So this is a sort of zoomed in image of a cell on a 50 micron fibronectin circle. And it's basically projecting about a 10 micron protrusion out over the non-adhesive area. But the LifeAct signal was just so striking to us. I mean, there's this obviously amazing retrograde actin flow going on, lots of bundles of actin and all that. Very, very different than what LifeAct looks like on a uniform fibronectin surface. I mean, this taught us right off the bat that all protrusions are not the same and that we really need to think about the context of them. So one of the questions we had right away with our ARP23 knock-in cells was, what does the ARP23 look like? I've already shown you the movie on uniform fibronectin. Much to our surprise, these spillover protrusions have virtually no concentrated ARP23 at the edge at all. It's there, it's flowing backwards by retrograde flow, but by no means is it concentrated at the edge at all in these spillover protrusions. So we thought, well, maybe the spillover protrusions are just some weird artifact, but let's put them on an adhesive surface, but one that doesn't engage integrins. And so what we saw was that, again, when we zoom in, we get what we expect for the fibronectin surfaces. We get this nice, lush, concentrated band of ARP23 at the edge, which almost certainly reflects that dense branched actin network. But if we just plate cells on polylysine, we get a strikingly different pattern for ARP23. Again, it is clearly moving backwards by retrograde flow, but it is not concentrated at the very edge in the same way at all. There's something very different when cells are engaging integrins versus not when it comes to this core component of the lamellipodia structure. So we can do chymographs of this, and as you might expect, the polylysine cells have much faster retrograde actin flow. And again, you really can see how different the leading edge looks, at least in terms of ARP23. Now, using that patterning, we can also put cells on dots of fibronectin, this is one of my favorite experiments that we've done. When you watch the movie, the cells spread out, and we can actually watch them as they encounter a new dot of fibronectin. They actually rip the old dots up, which is kind of cool. But it's a little bit hard to see the ARP. They're making sort of decently looking mature focal adhesions on the dots. That's is marked by VASP. But again, in these non-adhesive areas between the dots, we don't see a whole lot of concentrated ARP23, except when you look very, very carefully, and I'll show you this with some still images, when cells encounter new dots of fibronectin, they race right over them, and we see concentrated ARP23, which again, I think reflects the fact that integrin engagement is a really potent stimulator of ARP23 branched actin. And again, as they pass over that dot and then protrude do more of the spillover type protrusion into the zone between the dots, again, the ARP23 is not nearly as concentrated in those areas. So really, this leaves us to the question, why does integrin engagement so drastically change ARP23 dynamics? And so again, I'll sort of remind you that we've done this Haptotaxis screen to look for various factors, and FAX, SARC, Kindlin, ROGEFs, RAC, WAVE, ARP23, all required for Haptotaxis. And so the fact that all of these things co-occur in nascent adhesions at the leading edge of cells indicates that there's something really, really potent about this. And so we wanted to probe using some of our tools. Well, is it just that on polylysine you're not getting sufficient activation of RAC, or are some of the components not localizing properly? So I'll show you two quick examples of that. The first is going back to this optogenetic activation of endogenous RAC using the DHPH trick. And again, it's really quite lovely. We can image, in this case, just LifeAct that's tagged with MIRFP. When we trigger the recruitment of the DHPH domain and activation of the RAC, we get a really lovely lamellipodia that lasts for quite some time. And as you can see, marked very nicely there. But if we do that, that's on fibronectin. If we do exactly the same experiment now on polylysine, we basically get no change in the actin at all. So RAC activation is not really sufficient. There's some interesting stuff going on in the cell body, we think, that really reflects the activation of RAC. But at least at the leading edge, there's no slowing down of the actin, no concentrating of it in branched actin. It doesn't look like activation of RAC alone is sufficient to recapitulate what integrin engagement was doing. So the other possibility is that, well, maybe like something like the wave complex is just not localizing correctly if it's not on fibronectin. And we have a nice tagged wave 1, and it localizes absolutely beautifully to the leading edge, but it also localizes pretty beautifully to the leading edge on polylysine. So it doesn't seem to be that the wave is missing. So it's there, and presumably when we were activating endogenous RAC, it was activating, that's supposed to be sufficient to activate the wave complex and branched actin formation, but it apparently wasn't. There's something else missing that we don't understand yet. So it's not lack of wave, it's not RAC by itself, and so we're probing some other parts of this pathway as we speak with some other experiments, like activating the FAC-SARC pathway. But one other interesting possibility arose in some, again, we all had lots of time to do Zoom calls, and we've had some interesting conversations with people in the field. And I was talking to Doug Mullins one day, and he pointed me back to this paper that he published with Dan Fletcher, in which he basically did a very interesting in vitro experiment where he polymerized branched actin on an AFM cantilever where he could apply force to the actin network. And one of the things he observed is that branched actin networks increase in density when they're under load. And so what that led us to is the idea that maybe integrin engagement is simply the molecular clutch for actin, and it basically provides resistance and produces more load at the leading edge. And it's just all mechanics. So, what we're doing now is to try and, I'm just going to kind of walk you through some of the conclusions and then talk about some of the future directions here. So first off, ARP is clearly required to make lamellipodia and for making normal phagocytic cups. It's totally required for ECM haptotaxis. It's really required for efficient integrin-based phagocytosis. And obviously the structure of the lamellipodia is really profoundly affected by the substrate over which they protrude, particularly the dynamics of the ARP2/3 complex. It seems very, very sensitive to the substrate. And so, at the end of the day, the answer to my initial question is, what are lamellipodia for anyway? They're super sophisticated integrin delivery vehicles that are uniquely sensitive to substrate conditions, really, is what we think is going on. They're basically integrin sensors that allow cells to really probe their environment in interesting ways. And if you think about the nature of haptotaxis, different from chemotaxis, the signal comes to you by diffusion. You don't actually have to move towards it. In haptotaxis, you have to go to the signal. The signal is bound to the surface. And so you need some kind of protrusion. In this case, it looks like you need an ARP2/3 based protrusion in order to sort of really sense that kind of signal. So ongoing things that we're still working on, we're continuing to probe this integrin signaling, either the FAC-SARC, and test whether it's really a combination of RAC plus SARC that's required to fully activate ARP2/3. We're using some optogenetic constructs to try and probe that. We're also really trying to test the mechanical clutch hypothesis using optogenetic integrins or optointegrins, which are out there. And really what we ultimately like to do is to try and separate the mechanical aspects of integrin engagement from the signaling pathways. And one of the things we're attempting, which I think is probably a little audacious, but we're trying to build a fully synthetic clutch, which would mechanically arrest actin flow, but wouldn't induce any signaling pathways, although obviously we'll have to test that. But what I wanted to do now for this talk was, because you guys all care about cell migration, I want to sort of throw out some open questions for the field, and some of which I have data for, some of which I have absolutely zero data for. And maybe this is the ultimate pandemic thing, because we've all been in these Zoom calls and had lots of time to think and read and stuff like that. So I want to point out some things I think that are still very poorly understood in lamellipodia and cell migration biology. Okay, the first stuff is, how does this stuff get back to the front, right? So what is this arrow, right? So it's been in the Mullins and Pollard cartoon for decades now. It's basically saying all the components have to recycle their way back to the leading edge. This is really technically known as anterior grade transport. And there are several, people have thought about this and even modeled it in some ways. And so there's a couple possibilities for how it works. The first is simply diffusion, right? That there's basically the stuff can diffuse back to the leading edge fast enough. Seems a little surprising that it would be that efficient, considering you have this sort of kelp forest of branched actin moving backwards in the opposite direction. But one thing that we do know about it is that diffusion is size sensitive, right? Motor-driven transport. There have been people that have proposed that there are motors. Well, what are the motors? What is the cargo signal that gets all these various diverse cargoes transported back to the leading edge? Again, I have to say, I'm a little skeptical of the motor-driven transport. The other possibility is fluid phase transport, some kind of active mechanism there. And in a related sense, there are very intriguing observations about aquaporins or water channels, which are localized at the leading edge. They may be working with the fluid phase transport to basically get stuff back there. But it turns out there's a pretty slick way of measuring this anterior grade transport. It's a little bit indirect, but you'll see when we plated one of our cells on a square fibronectin pattern and we bleached a region of a corner, something really kind of amazing happened. And I'm going to let this movie play a couple times and just let you kind of digest it before I kind of give it away. Does everybody see the little faint dark band? Not the big band that's bleached, but the little faint band. We think that that represents dark bleached actin, which has been transported back to the leading edge and incorporated into the network and is moving backwards. So we can actually measure indirectly anterior grade transport by looking at the thickness of that little thin dark band. That represents how much dark actin got back to the leading edge and got incorporated. So we had an interesting opportunity to test one of these models, right? So we have cells that express tagged ARP23. In fact, it's tagged ARP23 complex, which is 280 kilodaltons. And those same cells can, in this case, express GFP actin. And we did a similar kind of bleaching experiment. And it's a little hard to see. I'll let you watch it a couple times. But in both the ARP23 channel and the actin channel, as shown in this little yellow oval, they get the same little dark band. So what that implies is even though they're 10 times different in molecular weight, they're basically anterior grade transporting at the same rate. So I think the diffusion hypothesis is a little bit shaky at this point. And again, I don't have an answer to this unanswered question, but I think it's really interesting. I think we've been engaging with some modelers to sort of think about the different parameters of what a transport mechanism would have to look like. And we don't yet have an answer, but I think this is a fascinating aspect of lamellipodia biology that is yet to be explored. So the next unanswered question is, how did ARP and non-ARP pathways compete and collaborate? I think that this is, people have worked on this, but I think it's, people sort of work on either non-ARP pathways or ARP pathways. We have an interesting opportunity to sort of begin to dissect the various contributions of these pathways. And so we started building cell lines with a whole bunch of different things tagged in different colors and started looking under some specific conditions. And I'll take you through a couple of those. The first is that we've started to use low doses of actin blocking drugs, in this case, cytokinesin D. And this is a result that is familiar to me from my postdoc days in Frank Gertler's lab, and that is that it looks like ENA-VAS proteins are uniquely sensitive to cytokinesin. And so this is an image of wave 1 and VAS, which are actually reported to be in a complex together. As soon as you hit the cells with cytokinesin, the VAS disappears from the edge and the wave complex is still out there, right? So yeah, this is not surprising. This is exactly what we saw years ago in Frank's lab. But we've recently gotten, really keying off of some beautiful work by Clemens Rotner, this molecule, this form in FMNL2, which does target the leading edge absolutely beautifully. And you can see we've started to make movies of it relative to ARP23 and to LifeAct. And we think this is going to be a very useful combination of markers to begin to dissect this. But we did a very simple experiment. VASP is chased off the leading edge. What happens to formins? So when we hit these cells with cytokinesin, in fact, the leading edge arrests, but the formin accumulates at the leading edge and intensifies. So these two actin polymerase / anti-cappers are behaving very, very differently with exactly the same drug treatment. We have a lot more work to do. So this is stepping up between 20 and 40 nanomolar cytokinesin. So it's a very potent response that's going on there. No idea why or how this works, but clearly it's different than ENA-VAS. So I wasn't completely transparent about our scarlet knock-ins. We actually knocked in more than just M scarlet into the ARPC2 locus. We actually also knocked in half of an optogenetic switch, in this case, half of the eyelid switch called SSPB. SSPB binds very tightly to the SSRA peptide, which can be incorporated into the love domain. And what we've done now is targeted this love domain to the mitochondria. And we were hoping, it's a bit of a failed experiment, that we could sequester all the ARP2/3 and then release it. And it kind of partially worked, but it's interesting. So we can suck away all the cytoplasmic ARP to the mitochondria very readily. But what you can see is that there's still plenty of ARP out at the leading edge. And part of this is we believe that we can't just express a high enough level of the mito eyelid. But it also says that, even though we've introduced an incredibly high affinity binding site on the mitochondria, it's not really sufficient to outcompete the stuff at the leading edge. Stuff at the leading edge is very, very potent. It's very resistant to sequestration. But it gave us an interesting idea. What if we did this experiment on polylysine? So we also don't fully sequester it, but something interesting happens to the actin in this situation. When we release it there and ARP goes back to the leading edge, the actin starts to look very different. When it's sequestered, we get many, many more microspikes. So sort of looking at stills during the sequestration to the mitochondria, the number of microspikes increases quite a bit. When we release it, the ARP2/3 from the mitochondria, it goes back to the leading edge. Now all of a sudden we start getting these actin arcs. So it truly is that actin is being, it is in balance between ARP2/3 branches and these other non-ARP2/3 pathways. And so again, we're hoping to use tricks like careful pharmacological treatments, as well as things like optogenetics to basically tease apart what's going on there. Now, of course, we have a knockout, and we can start looking at things like non-ARP pathways when there's no ARP2/3 around. And so, one of the things we saw right away is that wave 1 is localized pretty much okay to the leading edge, and maybe even along the shafts of Fibroblast in the ARP knockout cells. So we don't yet know whether that wave has actual independent ARP2/3 independent function, something that we're testing. But we can begin looking at least the localization, the dynamics, and ultimately the function of these non-ARP and sometimes upstream of ARP pathways in these ARP knockout cells. VASP, as one might expect, is sort of supercharged without ARP2/3. I think it has a lot more actin available to it. Interestingly, the Fibroblast localization, which normally is sort of fairly tip specific now, is a bit more all along the Fibroblast shaft, as well as at focal adhesions where we expect it to be. So again, we're beginning to look at a function. We had previously done an experiment where we sequestered enovasc proteins in ARP knockout cells, and they were extremely sick, where there was very little actin being made. But we need to return to those experiments now, I think, with more sophisticated methods like CRISPR knockouts. So I mentioned at the beginning of the talk that this 4-step cycle may not be completely helpful, right? In fact, these are not sequential steps, right? These steps are going on simultaneously. It's not actually a cycle, okay? Calling it a cycle is a bit like training wheels. And, you need training wheels to learn how to ride a bike. And I think the field has needed this as a sort of intellectual construct to think about it. But we have better experiments now. And I think we really need to think about this as a holistic process where these steps are actually influencing each other quite a bit. And, saying I work on leading edge, which is, admittedly what I've done for many years, without thinking about the other steps, I think is increasingly not going to be something that we can, support over the long haul. And again, I'm hoping at the end, I see that there's some stuff popping up in the chat. I'm deliberately ignoring that now. I apologize, because I need to get through the talk, and then we can have a discussion. I realize this is a bit of a provocative statement, but I think we need to think about this in a more nuanced and sophisticated way than just a 4 sequential step cycle. So the last thing, which honestly may be the most difficult, is, we have a big timescale problem in cell motility and really in all of cell biology, right? So I've told you about things happening on the seconds timescale, which is differential actin dynamics. You could even go to the microseconds timescale and look at protein conformation changes. And then we measure things that happen at 12 and 16 hours, right? Cell migration. And we can't, my lab, at least, and I suspect other labs, are having a really hard time relating things happening at these different timescales to each other. it creates huge experimental challenges. You can either sort of sample fast for a short time or slow for a long time, but doing both at the same time is really challenging. And I think, honestly, one path forward here is computational approaches and quantitative modeling. And so right now we're engaging with colleagues here at UNC and elsewhere to begin looking at how we can build models that incorporate these different timescales, because I think that's going to be really important if we want to understand the ultimate results of migration, like metastasis and other, higher order processes, things that are happening on the tissue scale. So just to acknowledge a few people in my lab, Mitch Butler is the one that made the ARPC2 knock-in cell line and has done some really beautiful imaging with it since then, and is working on this ARP matrix project. And some recent alumni which have contributed some data. I have some wonderful colleagues here at UNC, Tim Elston is a modeler, and Brian Coleman, who developed the eyelid system, and Jason Hawk, my longtime collaborator and friend from NC State, who is helping us with both experiments and modeling, and of course, the funding that helped pay for it all. So I'm going to stop sharing and see what kind of flaming I got in the chat.
Thanks.
So.
That was great, Jim. Thank you so much. So what we'll do, actually, we will read the comments and the questions out to you. We'll also bring in any comments or questions.
Thank you. Yeah, that would be most helpful, because I can't read them all at the same time. Putting links in and all that.
We'll take care of that. And also people watching live on YouTube might be popping in questions as well.
Sure.
So we're just going to go top to bottom. And also for everybody in the audience, if anyone wants to ask the question themselves, just do the raise hand function or say question in the chat and we'll get to you. So first from Claire Waterman, a couple comments. She agrees 100% with the purpose of ARP23 in the plasma membrane, that it's there to lay down integrins. And she also pointed out that Graham Dunn published on the rapid transport of the photo bleached actin to the leading edge and yes, cited a link with that. Yes. So that was the comment there, if you want to respond to that.
No, I'm aware of Graham's paper, and it was sort of sensitive to myosin inhibitors, but unclear exactly what the mechanism is from that paper. But yeah, no, there's some interesting older papers on anterior grade transport that we've been reading and thinking about. I don't think anybody's looked at anterior grade transport of other components like the ARP23 that we're aware of.
Okay, great.
Sam Lord says that Dyke's recent review argues against pure diffusion from solution, instead emphasizing the reservoir of membrane-bound actin monomers.
Yes. Yeah, no, Dyke and I have had many conversations about this. I actually teach some of the papers that sort of came from that, were the basis of that review. Yeah, so his argument, and I think there's some merit to it, is that there is a reservoir of monomers bound to various factors like the wave complex at the leading edge, which are contributing rather than them getting there. Although they still have to get from the cell body to the leading edge to bind to be that reservoir. So you still have a bit of a transport recycling problem.
Great, thanks. From Rina Kumari says, wonderful. Do you think one should also look at the endocytosis at the site of adhesion formation and branched actin accumulation? Yeah,
that's a great question. That's exactly I have a new student who's tackling this now, we've tagged Clathrin, and we're really beginning to look very carefully at the endocytosis pathway. There's some sort of really interesting changes in the endocytosis pathway that we're not really ready to talk about yet, but I think it's, I totally agree. And this is actually kind of goes to my point of, we can't sort of have, a stovepipe of cell biology. Like, I work on X process and I'm going to kind of ignore all the things that might influence it indirectly. And I think we need to be more nuanced in our thinking.
Thanks. Shashank, do you want to unmute yourself?
Thanks, Jim, for the wonderful talk, thought-provoking talk. One thing what kind of got me thinking a little bit, and it kind of goes against most of our own work about depolymerization and how important it is, the fact that you're seeing actin diffuse away or move at the same rate as a much bigger molecule ARP23, is there any chance that might argue towards the fact that these are not, severing is the main thing, and these small filaments that are actually being moved around and being added and removed rather than just monomers. And that again goes completely against what we've.
Yeah. So I think it was David Drubin's lab that had some interesting work years ago on actin oligomers, so short segments of actin actually being able to anneal onto barbed ends and basically getting non-monomer based barbed end growth. So it's possible. When we look at GFP actin, we assume that we're talking about sort of monomers, but that's not, we don't strictly know that.
Okay, great. Thanks. From Helen Hay, she says, thank you so much for the great talk. Would knocking down talin cause the same defect in lamellipodia formation for cells plated on fibronectin, since talin is essential for sensing the mechanics?
Yeah, that's a great question. We did some work with talin. It's hard because they just don't attach without it very well, so it's hard to do it. What we did do is knock down kindlin 2, which is the dominant isoform of kindlin in fibroblasts. And we basically completely lost lamellipodia and completely lost haptotaxic response. So we'd like to do it with talin as well, but it becomes a really challenging experiment to do. I think we'd have to do it, with Optogenetics or something else, because if you can't basically attach the cell, you can't begin to ask these questions.
Rob Insol says, someone, can't remember who, but it might be Tim Mitchison and Guillaume Shiraz, proposed that the lamellipod was a poroelastic structure with monomers flowing down the pores. That's faster than diffusion. What do you think?
Yeah, it's possible. I mean, again, I'm literally throwing these unanswered questions out without answers. I don't know the answer. I just want to challenge people to think about that, because I don't see a lot of work on it. So what you're saying is that, in fact, there are pores in the branched actin, which is moving backwards in the wrong direction, and that might lead to faster than diffusion. Yeah, but what about larger objects like the ARP23 complex itself? How would that interact with those pores?
Well, the pores are macroscopic, Jim. So if the retrograde flow causes a pressure gradient, then fluid containing all sorts of large stuff will get driven down the pores, and the limit will be way, way bigger than diffusion.
Okay. Yeah, interesting. I'll have to think about that.
For the data you're saying, that's quite an agreeable and compelling answer. I'll sign off here. It's your talk.
No, it's all right. I'm happy to have. I was hoping to engage lots of people in this. Yeah, I think that would if it was fat, would that I guess my question is, would that mechanism still be size sensitive? I don't know. So.
Okay, great. Sounds like good open question there. From the next one, from Joanna Avaska, says, Robert Gross has shown that FMLN2 binds directly to the integrin alpha tails. Do you think that the integrins are maintaining or concentrating the FMLN2 on the membrane in your cytoclasin experiments?
I have no idea. I hadn't thought about that as a possibility. That's interesting. I guess how would you envision cytoclasin changing the integrin tails exactly that would increase the binding of FMNL2? Because there seemed to be sort of an enrichment of FMNL2 upon low dose cytoclasin. I don't know. Yeah, that interesting question.
Tony Tsai says, thanks for the great talk. Is there anything special about the geometry of lamellipodia that makes it suitable for the mechanical response, while Filopodia can't? Or is the mechanical response something special about ARP23 and lamellipodia is just the end product of ARP23?
Yeah, this gets to our conundrum, right? Is this a uniqueness of the upstream signaling into ARP23? Is it about the substrate geometry of a sheet versus a finger? I think it's maybe a bit of both. It'd be my guess. I don't have data to support that, but clearly there's a uniqueness to the upstream signaling that, whatever's upstream of Filopodia isn't as responsive to integrin engagement and ECM content. But I also think that the unique sheet-like geometry of lamellipodia, which by the way are also sheet-like when they're in 3 dimensions. So there's something intrinsically sheet-like about lamellipodia, and I debated about whether to have that as an unanswered question. I don't think we fully understand that answer either. We thought it was just the flatness of 2D cell culture for a while, but now with new light sheet data, it looks like you just make flat sheets with ARP23 in it. I don't know. Yeah, interesting question.
Great. From Tina Tootle, she says, it seems like there was really dynamic ARPC2 scarlet in the nucleus. Have you looked into nuclear defects in the mutants?
Yeah, we've tried. There's quite a bit less inside the nucleus. I think some of those movies are a little bit deceptive. Most of what you're seeing is sort of either above or below the nucleus. We've done some more careful sectioning, and there's some inside the nucleus, but no, we honestly haven't. We don't keep, we haven't. That's just, again, there's almost too much for us to tackle at this point. There are some reports in the literature using actually some of our cells that ARP23 and I guess it's NWASP are in the nucleus and functioning in some DNA repair pathways, but we haven't gotten very far looking at that.
Claire Waterman says, who or what protrudes the cell edge in the absence of ARP23?
So our assumption is that you essentially make abundant filopodia using some combination of enivasp, formins, FASN, but there are interesting bundles of actin that sort of connect between the filopodia that lie parallel to the leading edge. And when we treat ARP23, I think we only did it with the knockdown cells. We treat those cells with blebbistatin, we actually get more filopodia as if they were limiting things. And that's our guess is that this is protrusion that's polymerase based.
Yeah, I mean, there's sheets of membrane that are sort of filling in between those filopodia that, something's got to be at the leading edge, uncapping or promoting.
Yeah, again,
do you know the molecules?
There's really abundant enivasp and formins that look like they're super active in the ARP knockout cells. And our previous work, we knocked down profilin, and which we think provides sort of the main profilin actin fuel for those polymerases, and that really did a number on the actin structures in the ARP knockout cells specifically.
All right, great. From AJ Kitness asks, can lamellipodia only form in the context of an ECM-bound substrate? Or can adhesion that is cadherin dependent also determine lamellipodia formation? If lamellipodia are...
Go ahead. No, I was just going to say that's a great question. Sorry, go ahead.
If lamellipodia are seen in the context of cadherin interactions, is it more likely that it is not the cadherin interaction that's responsible, but an associated ECM component?
Yeah, I don't know the answer to that. James Nelson had some fabulous experiments years ago where he could plate cells on synthetic substrates which were engaging cadherins. I don't know what would happen to ARP23 and lamellipodia under those circumstances. That would be really cool because that actually may help us to tease apart some of the mechanics versus the signaling. I don't know.
Thanks.
Okay.
Thanks, AJ. Adam, do you want to try to parse through this whole discussion that's happening? There's been more in the chat.
I'm ignoring the chat. I can't deal with the chat. Sorry.
A lot of back and forth about this poroelasticity argument.
Okay.
I'll just mention Rob Insol's comments. It was a paper. It was the Dunn paper that says retrograde flow makes a pressure gradient, and that monomers and other things are therefore driven forwards through the pores.
Yeah, so that would be a sort of a fluid phase transport mechanism, which I tend to agree makes the most sense. But exactly, does that work together with an aquaporin at the leading edge? That's the drain at the bottom of the bathtub letting water out, but not these, other things. Does it work together with that pressure gradient? Exactly what generates the pressure gradient? Is it dependent on the cell type? I mean, there's a lot of, yes, I agree. That's probably the most likely explanation.
All right, and Rob's follow-up comment to that is aquaporin as a drain in the bathtub.
Yes, Peter Agre would be very happy. I agree. We literally have just during the pandemic, we couldn't get into the lab, so we just did a lot of Zoom talks and reading, and it's been kind of interesting, and we've revisited some of these really old questions.
Yeah, maybe we'll see papers sort of do a dip and then an increase, right? That'll be interesting. One question from me, I'm just wondering about how substrate stiffness changes any of these responses. Is it, is it just sort of a gradient of response, or do you get massive flips and totally different behaviors on softer
we're
substrates?
just setting up those experiments. It's a little bit more difficult to do really nice imaging up on top of soft gels, but we're sort of slowly working our way through it. Really what we'd like to do, obviously the stiffness is going to impact, integrin engagement and signaling. So I would expect that there would be some related phenomenon in substrate stiffness versus ECM content on glass. What we'd really like to do is basically make counter gradients of stiffness and ECM content and sort of see which one wins. And again, we're just gearing up to do those kinds of experiments. But I think that would be very exciting.
I like the contest idea. We have a question from Michael Way. Asks, is there any
birthday,
evidence?
Michael.
Happy birthday.
It's Michael's birthday. I saw it on Twitter. Happy birthday, Michael.
Well, hopefully he wanted everyone to know. So
he put it on Twitter. I'm assuming he did.
It's true. It's public knowledge. So is there any evidence that ARP23 interacts with lipids, which might constrain the plane of new actin filament growth?
Cool idea. I don't know. I mean, the planar nature of branched actin protrusions is fascinating to me. And again, I don't understand it. I think Ryan Weiner has some interesting data on membrane curvature, helping to target things like the wave complex. So it could be that the NPFs are constrained to be concentrated in certain places by curvature. Lipids could also be a part of the equation as well, but I don't know.
I've got one more from Sabag, who's asking if you've looked at the microtubule organization and dynamics in your ARP23 knockout cells.
Yeah, one of my former students, Emma Wu, who's in Beijing now, has published a paper on this, and it does look like the microtubule network is different, and it looks like it's there's some differences in the acetylated microtubule population. We haven't really worked on that. I sort of, again, I have to limit my appetites for starting new whole projects but this cell line has just opened a whole bunch of stuff up that we haven't had a chance to tackle.
Okay, great. From Fred Fregoso, says, great talk. Do you have any thoughts on how inhibitors of ARP23 like ARPin or coronin influence haptotaxis?
Yeah, it looks like, I didn't show the data and we haven't published it yet, but it looks like coronin 1B, 1C double knockouts are fairly defective at haptotaxis, and it's because the lamellipodia are not tuned properly. We've also seen that GMF knockdown cells are defective at haptotaxis. We've published that. Basically, anything that affects the cycling of lamellipodia seems to have an impact on this haptotactic response. It's exquisitely sensitive to things that mess up the lamellipodia like that. We haven't looked at ARPin specifically, but I can speak to the coronins and the GMF, and they're clearly affecting it.
Thanks. And Anna Pasipera says, have you tried activating integrins with manganese in cells plated on circles with polylysine and looked at the ARP23 localization?
Yeah, we've talked about those experiments and we've tried some of them. I don't remember the results. I don't think anything super dramatic happened, but again, it was hard to know that we were activating the integrins. We were trying to get like the staining for the active integrin antibodies to work well. And it was, we sort of chased our tails a little bit. But it's a good experiment. We'd like to do things like that in the future.
Okay, great. So that's sort of what we have in the chat, but people should feel free to raise their hands or pop more things or unmute themselves to ask more questions as we're sort of wrapping up. Sorry. And one last question from me, and maybe I might have missed a little bit. I'm used to thinking about cells as entities and sort of collectives of cells, but I was really struck by your experiment where you did the photo bleaching and then you had that thin line. And the thing that was so striking to me about it was how sharp that line was. So it means that whatever process is moving that actin forward is happening, that's a really wide width that it's happening very spatially. It's
almost 10 microns.
Very consistent, right? And so that length, it's all moving at exactly the same rate, or else it wouldn't be such a sharp line. And so I was just wondering if you had any thoughts about.
I showed the movie because I think it's super cool, and I still don't understand it. I was hoping the collective wisdom of the cell migration seminars could help.
Yeah, I mean, there's something there that we can tell a cue from the sharpness, I think.
Well, again, Jason Hawk, who's a chemical engineer by training and teaches transport phenomenon, has been sort of puzzling around with this. We've shown them this movie like a million times, and it's still not exactly clear how it's working. Alex McGilder had some wonderful modeling, and he was like, I remember at the end of the discussion section, he's like, diffusion might just be enough to take care of actin, but anything bigger, all bets are off. So something interesting is going on there. I don't know.
All right, thanks so much. Sam, you are next. You had your hand up.
Hi, I just want to follow up a little bit about the membrane-bound reservoir, and I like to think of it as like a field of grass, like and the barbed ends of the lamellipodia being like cows grazing in the field of grass, and they might deplete a small region, but then they can move to another part. So as the membrane moves, or as the lamellipodia moves, it can encounter new pre-bound actin to the membrane. So it doesn't necessarily need to be limited by diffusion. It's now limited by the flow of the membrane and the movement of the barbed ends to new parts of the membrane. Of course, that doesn't explain the ARC2/3 flow, but I just wanted to, I've been thinking about that cow analogy, and I just wanted to say it out loud.
I like it. Again, the sharpness of the bleached region returning is also sort of like, when you think about cows moving onto different blades of grass, is it that there's no way they can deplete all of it? I mean, there's lots of questions like that. And again, the ARC2/3 transport, I think, is interesting, that looks very similar to actin, right? So, but maybe it's just actin oligomers or something. I don't know. But yeah, I like that idea.
Fernando in the chat asks why you looked at formin like protein 2 and not other formins like dia2.
I've had lots of conversations with Clements about it, and I've read his papers very carefully, which are quite lovely. I think it's the most compellingly localized there, because the endogenous FMNL2 is clearly there. The tag stuff goes there very reliably. Whenever you express, we've not really seen, and I'm happily corrected if I'm wrong, but MDIA often has to be sort of modified where the DAD domain is clipped off to get reliable leading edge localization. In full length endogenous dia2 doesn't, as far as I can tell from reading the literature, localize quite as nicely to the leading edge. So I think it's the formin that it certainly in our hands works the way it does in Clement's papers, and seems really reliable marker for that formin, at least at leading edge.
Great. Are you okay to stay a couple more minutes, Jim?
I'm happy. This is like the most extensive qualifying exam I've had in 20 years. So I'm ready for it.
Well, I think you're passing with flying colors.
Well, I will see what the committee says when I leave the room.
Well, we always go over time, and I thought we were gonna, you left enough time for once. But it's great that people are so excited. So yeah, I think Michael Way was following up on what you were just mentioning, and says Clements rather in his 2008 EMBO paper. Okay, I'm not sure what this is. It looks like a typo. Says the ARP2/3 complex interactions and actin network turnover in lamellipodia, specifically in figure 1C, also sees the same dark line after photobleaching.
I'll have to take a look at that, Michael. That's a good catch.
Great.
And Kai Merck says neurons are regularly plated on polyalysine in contrast to most non-neuronal cells. Could many differences in the movement of neuronal and non-neuronal cells just be explained by the choice of different substrates?
I think that would be very interesting. Yeah, I'm not sure historically why. I'm aware that polyornithine and polylysine are sort of typical substrates for neurons. I don't know why they don't plate them on laminin. They certainly love laminin, right? But whether their movement or their actin dynamics would change based on that, I don't know. My colleague Stephanie Gupton here at UNC and I have had several conversations about this, and I think it would be interesting to look at.
All right, great. And then I think maybe the last thing we have is just Michael following up that the 2008 EMBO paper was GFP tagged actin.
Yeah, so was mine. So was ours.
Okay.
Yeah, but we were able to sort of track ARP and actin with 2 different colors. I think that's the difference.
All right, and one more question coming in from Janice Burkhart, who says, in migrating T cells, we see very different actin responses depending on whether the cells are migrating on beta-1 or beta-2 integrin ligands. Have you looked at beta-2?
I think I saw this paper from you, Janice, and I think it's fascinating. We have RNA-seq on our fibroblasts. It doesn't look like to express any beta-2 that I can remember. I think that's a beta subunit that may be sort of hemopoietic specific, but I couldn't swear to that. We've thought about trying to tackle it from the integrin perspective, and that's certainly how the field has sort of done it with, specific inhibitors like selingitide, which I'm not sure I still fully understand how it works, and some other things. So yeah, cool. I think that there's some really fascinating connections between integrins and actin, specifically ARP23 actin, that we've just scratched the surface of.
Great. So I think we are all done in the chat. So in that case, Jim, thank you very much for a wonderful talk and a great discussion.
Thank you all for jumping on another dreaded Zoom call, but I appreciate all the questions and all the feedback.
This is my favorite Zoom call of the week.
This is your favorite Zoom? Yeah, that would not be hard to beat the other ones, but yeah, no, it's awesome. Thanks everybody, and thanks for the 2 of you for hosting. Really fun. I had a really good time.
Thank you, Joe.